Well, good morning, everyone. And we're talking to Behzad from Iran this morning. One of the great delights about this is being able to talk to people from around the world and scientists from around the world. Now, Behzad, would you like to introduce yourself for us, please?
Thank you for having me, John. My name is Behzad Khansarinejad. I'm an associate professor of medical virology at Iraq University of Medical Sciences in Iraq, Iran, and I'm also a virology lab director.
Right. So you've been involved in this from the start of the pandemic, Behzad?
Yeah, from the first day that the first case in Iran was recognized. The first case was recognized in Tehran, and the second case was recognized in Iraq in our laboratory.
Wow. Right. So you're right at the start. So give us a bit of a history of the pandemic, if you don't mind, in Iraq. We're going back to January 2020, something like that.
It was almost, yes, January 2020, and the first case was recognized in a city, Qom, which is near Tehran, and then afterwards we reported a lot of cases from parts of the country, from north of Iran, center, south of Iran, and we involved in the pandemic, and we have passed 5 waves, most of them when the vaccination wasn't started yet, but now actually, at present, the number of cases and the number of mortality has reduced. For almost late August, it was a trend downward,
So was there a lot of deaths at the start of the pandemic in the early waves before the vaccination was instigated?
Yeah, it was very disastrous. We had 5 disastrous waves with a lot of deaths. The confirmed cases of this was near to 700 a day, but we assume that the actual number could have been more. We guess that it was twice the actual number was twice the confirmed case. Yes.
So the infections were clearly much greater than the cases, and did this spread all around? These waves spread all around Iran in the rural areas as well as the cities.
Yes. From north to west, from north to south, south west to east. Yes, all around Europe.
Yeah, and tell us about your involvement in the virology. When did you first get? When did your lab first get assigned duties in this?
Yeah, it was almost January 2020, and we were prepared for that because before the cases are recognized, we were ready for that. Some laboratories in Iran, for some virology laboratories, had this meeting that to be prepared. We had tests, we had primer and probes, we had primers for sequencing, and in that time, influenza existed in Iran. We had a lot of cases with influenza negative, and we used to do a lot of SARS-CoV-2 tests, and finally we found that the virus has entered into the country. Okay. And as you can see, the number of confirmed cases, it's the latest data. And we have these 5 ways. The number of, these are daily new confirmed cases. And since the late of August, the number has been reducing up until now, that the number is about 2,000 cases a day, a little bit less than 2,000 cases a day. And the number started to becoming less than 5,000 when the vaccination rate in Iran was about 45% to 50%, actually. And accordingly, the number of deaths has reduced since late August, and now it's less than about 50 cases a day. We have these 5 waves of death, and as I said, the actual number was about twice as we calculated using excess death rate data.
So there's been quite a lot of deaths quite recently in Iran, then, as late as a few months ago.
And it was for Delta. It was for Delta wave, which was the most huge wave was the 5th one, which was related to Delta. And this positive rate of the test, and as you can see now at present that the number of cases has reduced, the positive rate of the test is quite good for Iran. Before the current time, especially at the peak of the 5th wave, We had this more than 53% for positive rate, and now it's less than 2, which
so
is
the
enough.
53% positivity means you had massive community transmission during that time.
The number of tests was not suitable, was not enough for that time, but now it's quite enough. And you might be interested to know that the number of influenza cases at present is much higher than the COVID cases. The positive rate for influenza is almost more than 30% in most of the country. In most provinces, 30 to 35% of samples become positive for influenza. And another 35 to 40% become positive for some common cold coronaviruses, the cousins for SARS-CoV-2, especially OC43 and HQ1 viruses.
These are the common cold viruses, the
cold
coronavirus,
yes,
common cold virus.
They are separated from the COVID-19 agent. And together, these viruses, influenza and common cold coronaviruses, are responsible for about 70% of the cases every day in Iran,
Why was the death rate so high during the Delta period when the vaccination rate was reasonably good?
Actually, we don't think it was reasonably good because in that time only 20% of the population was vaccinated, and vaccination started very late in Iran, but its speed was almost good. And when the rate of vaccination reached to 45 to 50%, the number has reduced. But we are not so sure that the decreasing the number was because of the only vaccine, because the population of Iran mostly infected with natural infection. We believe that almost 75% of the population generate immunity using natural infection, and the combination of natural infection and vaccination has resulted this situation that the virus can't spread very effectively. Omicron has entered into the country, has recognized almost 10 days ago, but the positive Omicron cases are still very low, around 40 cases in whole country. With the screening test that we have, which is not enough, but it's quite good, because we use some real-time PCR assay for screening the mutations, and when we find a case, the sample is sent to the reference laboratory in Pasteur Institute of Tehran to confirm by sequencing. Okay.
Yeah.
So, as I said, when we believe that most of the country generate immunity using natural immunity, and because of this natural immunity, the infection rate is reduced. We believe that the infection rate is reduced because of vaccination and natural immunity. I just need to show some illustration about the virus. As you can see, it's the S protein of the virus. It's the ligand of the virus that the virus used to attach to the cell receptor ACE2. And it is the S gene that is used for making next generation vaccine, mRNA-based or viral vector-based vaccines. But the virus has a lot of other proteins, M protein, which is the most abundant protein of the virus, and is also an immunogenic protein, which means immune system can recognize it and act against it. And N protein, your nucleocapsid proteins, E proteins, and the virus produce a lot of proteins in infected cells. So during natural infection, all these proteins elicit immunity and result in a polyclonal response to the virus. It's the genome of the virus. The genome is almost very big in comparison to other RNA viruses. Almost 2/3 of the genome comprise the region that makes a lot of enzymes, such as RNA polymerase and other enzymes, which is necessary for replication of the virus. And the other proteins like S, M, N, and E are located downwards. If you take S gene and look at that, it has a lot of epitopes, which means the parts of the antigen that immune system recognize and against them. And B cell epitopes are different from T cell epitopes. They might be in different places, and some of them might have overlap together. So when an antigen introduced into the cell, different parts of it elicit immune system and make immune response. And every single protein of the virus has these B cell and T cell epitopes. So when the whole virus enters to the cell and starts to replication, different part of immune system can be activated and act against the antigen. So in a natural infection, S gene is expressed, M gene is expressed, N gene expressed, and a lot of other proteins. More than 25 proteins is produced into the cell, and all of them can elicit immune system. So the result of this polyclonal response is a huge immunity with a longer memory and a stronger response. And that's why in many countries that had a lot of natural infection, the cases are reducing better than the other countries. But in the next generation vaccine, only the gene for S gene is located. And the logic behind that is that when S gene is produced into the cell, antibody response to the S gene neutralize the virus to attach to the cell receptor and inhibit that. But as you can see in Omicron and other variants, the sequence for the S gene has changed. For example, in Omicron, 32 amino acid has changed, and this neutralizing antibody might not be effective. So one of the question about the next generation vaccine is that why they are still using the S genes for wild type variant or classical variant to the vaccine and don't add the S gene for current variant of the time and other proteins to the vaccine to make a polyclonal response. I personally believe that the vaccines should be updated based on the situation that we are encountering now. Otherwise, the number of infected persons all around the world is going up. Yeah.
But what seems to happen is that even with the old-fashioned, what we might say, the old-fashioned type of antibodies, it's still generating a T cell immunity that seems to protect against severe disease.
Yeah, sure.
So why is it still protecting against severe disease if the neutralizing effect of the antibody is not working?
Yeah, that's quite true, because neutralizing is not the only mechanism that our immunity use against the virus. Neutralizing is only one of the several different mechanisms that is used. And the other mechanisms, such as cytotoxic T lymphocytes that act against the virus, can kick in and starts to clearing the virus, and other mechanisms. So neutralization is not the only response. That's why we need other proteins to be located into the vaccines, next generation vaccines, and that's why we need that the sequence of the S gene that is located into the vaccine change based on the variant of the time, you know.
Yeah, but given the vaccine stimulated the B and the T memory cells with the old wild type, why is it that the B and the T memory cells seem to be active against the Omicron, for example?
Because there are some sequence similarity between the variants. You know, only 32 amino acid has changed among hundreds of amino acids. So and the fact that the epitopes for B cells are different from epitopes for T cells. So some neutralization activity might not be effective again, but the T cell response might be effective. So vaccinated people are still have this lower chance for getting very bad disease, and the mortality rate with vaccination is reduced at the time. You know,
right. So on the spike protein, there's several hundred amino acids, and only 32 have changed.
Yeah.
So the T cells are sensitized to the other few hundred amino acid sequences, and that's what's preventing severe disease.
Yes.
But it could be optimized, as you say, much better if the vaccine was tuned to the modern Omicron.
That's really the case. And tuned with other genes. they can put other genes into the vaccine, and not just only based on the S gene. Other genes can be inserted to the plasmid that produce mRNA vaccine, or the plasmid, or a vector that resulted in a viral vector. So proteins like M, like N, we know for a long time that they are immunogenic. And the result of polyclonal response might be very effective to produce a better vaccine against the virus.
So with natural infection, we're getting M immune response, N immune response, S immune response, and about 20 others, in fact.
That's right.
So natural immunity is going to give a better quality of immunity than the specific S gene vaccine immunity. Is that sort of the thinking here?
We have always knew that. Actually, John, I don't remember any respiratory virus that if it has a vaccine, its vaccine is more effective and its memory is more than the natural infection. But it's not an anti-vax message.
No.
Yeah, because we need vaccine. If the vaccine has reduced the number of mortality that did that, it's a very good success. But when we are talking about the science, we have to talk about the reality, as you told before, and it's the reality. The immune system, the different hundreds of epitopes on a protein can produce a polyclonal response that their effect, their bonus effect, is more effective than only a directed response toward a special part of a protein or a special part of the virus,
And do you suspect that natural infection will have a greater longevity?
Yeah, I believe so. Yeah.
Do you think we might be talking years here, or I know it's an unfair question, but do you think it might be a long time?
Yeah, for most respiratory viruses and intestinal viruses, the immunity is not for whole life. You might infect it different times in your life, but the next time, normally the severity of the disease is less. For the first infection, normally in immunocompetent person are the most severe one, but when you infected after 2, 3, or n years from now, they are normally softer, less severe, unless the people is immunocompromised.
So the hope is that natural infection is going to generate a milder illness for years. We don't know how many years, but years into the future.
But yes, because when you see back and look at the evolution, we had a lot of these viruses that are existing now. Some of them did the same thing that SARS-CoV-2 when they were new and when they made an outbreak. But after that, because of this immunity, the whole immunity or herd immunity, they become softer. They become like influenza, like seasonal coronaviruses and other viruses around us. We have reached to an equilibrium with them, or immunity. The SARS-CoV-2 virus was the most, recent one, but it wasn't the most pathogenic one. We had experienced a lot of these infections before. Our ancestors experienced that, but in that time we didn't record that. We didn't have PCR. We didn't have this.
Sure. So humans have been getting exposed to coronaviruses for thousands of years.
Sure.
Yeah. Now, okay, so that's interesting about the longevity of the response. Now, the other question in my mind is, if we take a country like South Africa or a country like Iran, where, say, 75% of the immunity that is enjoyed by the population is as a result of natural exposure. And if we take a country like the UK, where most of the immunity is as a result of vaccine, or if we take a country like Australia, where all of the immunity virtually is as a result of vaccine, what are the implications of that for Omicron?
So actually, I always thought that the countries like Australia, New Zealand, and the countries that you mentioned will experience the infection, because these vaccines, the current vaccines for SARS-CoV-2 doesn't prevent infection. They might reduce the number of death and severe disease, but they don't prevent the infection, because the the immunity coverage of them is less than the threshold for herd immunity of the Omicron. So people get infected, but fortunately, because they are vaccinated, the number of deaths is going to be reduced, and the herd immunity is produced based on the combination of natural immunity and vaccine immunity. So
I think you've answered a critical question there, Behzad, actually, because my concern was that a country like Australia, where it's all vaccine-induced immunity, that they wouldn't be enjoying protection against severe disease from Omicron. But it looks like they will enjoy protection against severe disease, even though they're only sensitized to the spike protein, not the polyclonal response.
We have to see that. And one of the most interesting things about comparing countries together is that they have different situation at the special time, because Australia and South Africa are located in the southern hemisphere. And you can't easily compare a country that is in northern to southern, because now it's winter here. Yeah. There is a summer, and the level of UV and other parameters that define the severity is have to see what's going to happen. But personally, I'm very hopeful that the number of severe disease and death is going to be reduced in countries like Australia, because they have this level of good vaccination. I hope so, but this is a disaster.
Thank goodness for the other few hundred amino acids on the spike protein that the T cells are still sensitized to, which is going to prevent the severe illness and death. So basically, I think what you've said there is we're fairly hopeful that people in Australia or in the UK who've got protection against severe illness as a result of what we might call the old style vaccines, they can get exposed to Omicron. They're not going to get very ill. And am I right in saying that their exposure to the Omicron is going to give their immune system a really big boost?
Sure, definitely a really big boost, but it doesn't have still a guarantee that the person doesn't experience severe disease. But normally, for most cases, the likelihood of severity of the disease is reduced.
So this is really an ideal situation. Most of us are protected from severe disease, but we're still going to get natural immunity.
Yes.
On top of that, because we've been exposed to the natural Omicron variant. So this sounds like pretty good news, really.
Yeah, pretty good news. Yeah, but not a complete vaccine. I wish we had a better vaccine, like traditional vaccine, like measles vaccine, mumps vaccine, that when you use them, you don't get ill. But at the present, in this pandemic, with this short amount of time, we couldn't adapt ourselves very rapidly to make better vaccines.
Yeah. Now, most of the vaccines in Iran are actually not the mRNA and not the adenovirus vector. They're actually the attenuated or the dead virus. So in Iran, the vaccine given mostly has been the whole dead virus, the whole thing.
Actually, they are inactivated virus.
Inactivated, that's the word I'm looking for. Yeah,
sure.
Yeah.
Yes, almost 85 to 90% of the population has received this inactivated virus, Chinese Sinopharm or a domestic one, Barakat, which has whole particle, but the response in comparison to attenuated virus is still lower, because we know that inactivated viral vaccine, especially with that type of adjuvant or alum adjuvant that they use in vaccine, induce more humoral response to cellular response. And with this type of humoral response, the response is not as complete as a whole virus that is amplificate into the cell, attenuated virus. So most of the responses are humoral and antibody, and some of them are not neutralizing because during the process of inactivation, the structure of some proteins are changed, and the response is not as complete as the virus that replicate into our cells. We don't have any mRNA vaccine in our country, and almost 10% of the population or so has received DNA viral vector-based vaccines,
So we're left with a situation in Iran where most of the immunity, as you say, is as a result of the natural infection.
Yes, sure.
Induced by the previous waves.
Yeah.
So basically what we're saying is the inactivated vaccine, they're good for generating antibodies in the short term, but they're not so good for sensitizing the B and the T memory cells for the longer term immunity.
Sure, yeah, sure. And the type of antibody that is produced with inactivated virus are immunoglobulin G and immunoglobulin M, no immunoglobulin A. So the response and immune response is not as complete as the attenuated viral vaccines.
But that would be the same with the mRNA vaccines. They won't produce IgAs, will they?
Yeah, because they are introduced into their muscles,
Okay.
The root of infection is not respiratory,
So if you give an mRNA vaccine, you'll end up with immunoglobulin type A's in your mucous membranes.
Yeah, no, with mRNA vaccine, no. The level of immunoglobulin A is not very high at the respiratory areas.
Right. Yeah. That's what I thought. Yeah. So how do you see the situation in Iran now? Because I think, can we assume that Omicron is going to go all over Iran in the next few weeks?
We have to wait and see. I can't exactly predict, but I think because the level of natural immunity is higher in Iran, the speed of propagation for Omicron is gonna be lower than in comparison in many other countries that have immunity by vaccination. Almost 10 days ago, the Omicron cases was diagnosed in the country, but the number are still very low, and about 40 cases have been diagnosed. So I believe because of this buffering situation for viral propagation, the spread of the virus can be very limited in comparison to the time that the Delta was hit by the country,
Yeah. Interesting. But in the South Africa situation, where there was a lot of natural immunity, Omicron was reinfecting. Yeah. There was breakthrough infections from people that had a level of natural immunity, and they were getting mild disease, just not getting very sick.
It's quite possible here in Iran.
So it's possible that the virus could run.
Yeah. We have to see what's going to happen, but it is completely possible because. But another thing that I guess is that the level of natural immunity in Iran is even higher than South Africa.
Yes.
Almost 10% higher.
Yeah. Interesting. So, do you think Omicron in places like the UK is going to replace Delta? Is it?
Sure.
It's out competing it? So in the same way that Delta basically eliminated Alpha.
Yeah.
Is Omicron going to eliminate Delta?
Yeah, it's quite dependent on the R0 value of the Omicron. We don't know exactly the R0 value of the Omicron to compare with the R0 of Delta, but it seems that it's more infectious, and because it's more infectious, it finally displaced the Delta and become the dominant variant in UK and everywhere that it enters.
So this sounds really good news. If Omicron's causing mild disease and Delta causes severe disease, the Omicron is basically sweeping out.
Yeah, sure.
The Delta is getting rid of the Delta for us.
Kind of like an attenuated vaccine, It is not an attenuated vaccine, but can act like an attenuated vaccine. And John, one of my questions about the vaccine platforms is that why? Because I've studied virology, I know how they make vaccine. But I was not involved in the process of making vaccine. I'm more involved in diagnosis of the viruses. But my question is that why they are not still trying attenuated vaccines? They say it is dangerous, it's a pathogen virus, we can't easily be sure that it's safe. But we had attenuated vaccine for viruses that are more pathogenic than SARS-CoV-2. For example, yellow fever, measles. Measles is more pathogenic than SARS-CoV-2. When you see that the number of deaths because of measles is low, because the measles is an endemic virus, and we have herd immunity and vaccine immunity against it. But when as a new virus, when it caused outbreak in Fiji Island, 40% of mortality produced by measles virus. So we had attenuated vaccines against more pathogenic viruses.
Yeah.
So why don't we try attenuated platform for SARS-CoV-2? Because it's the nearest platform to actual viral infection, and it can result in a longer immunity and longer immune response.
I think people don't realize just what a dangerous disease measles is, and it still kills a lot of children around the world. It's a really serious illness if it's not prevented.
Yeah.
So basically, with the Sinopharm vaccine and the measles vaccine and the Iranian domestic vaccine, what people do is they brew up huge amounts of virus. And I presume they do that on a tissue that must have a cellular culture that they brew up the virus in. You end up with untold billions of viral particles, and then you attenuate them to stop them. Basically, you kill them off. You greatly weaken them. And then you inject these to develop this polyclonal immunity, which happens with measles. Now, the fact that the first generation of Sinopharm didn't work particularly well doesn't mean to say that this technology couldn't be developed to get a polyclonal attenuated vaccine. And the reason that is not done is really a good question. People seem to have put all their money in the mRNA technology, and to some extent in the adenovirus vector technology, while I think we could basically say in Western countries ignoring the tried, tested and proved vaccine technology of attenuating the whole virus. And the reason that is not done, I don't know the answer to that. Some people might suspect that it's commercial interests.
Yes.
Some people might suspect that. I wouldn't know, but some people might think that existing vaccine technologies are making a lot of money, so why experiment with another vaccine technology that you'd be going back to scratch on? But it would give a much more polyclonal response. So all we would need to do is get an attenuated vaccine which is good at stimulating the T cells and the B cells, in addition to the antibody immunity, and then we'd be onto a good thing.
All epitopes.
All epitopes, and the epitope is just the part of the antigen that the immune system recognizes as being foreign and generates a reaction to. That's a remarkably good question, and I don't think I have a scientific answer to that at the moment. So I'm optimistic that Omicron is basically going to be the end of this pandemic as it displaces Delta and it becomes an endemic common cold virus. Is this just wishful thinking on my part?
Yes, I think so. I have this belief, and I think after this huge wave in many countries, Western countries, in UK, in US, Canada, most of the population obtained this permissive herd immunity with combination of vaccine and Omicron variant, and the pandemic will come to its end, and the sporadic form of the disease or endemic form of the disease is going to be continued in next years,
Yeah, for the next 5-10 years or whatever. Yeah,
sure.
Yeah. So basically, Omicron is the best thing we could have hoped for. It's going to introduce massive herd immunity.
Yeah, because its pathogenicity is lower.
Yeah.
Its infectivity is higher, and most of the countries are vaccinated. Most of the involved countries are vaccinated, and it could be a very good news to finally reach to the end of this pandemic.
So this pandemic's going to end because nature has just evolved this new type. It's not human beings being clever. It's just the natural evolution that's built into the ecosystem of the planet almost.
That's really the case. That was a very good sentence, because we saw that still the natural immunity and natural event that has obtained us from getting ill and from getting extinct is working again.
Yeah,
it's hopeful.
So given that the Omicron is so infectious from all the data that we have, and given that it's causing mild disease, if another variant was to evolve, that would only become the most prevalent variant if it was more transmissible than Omicron.
I still think it depends to the immunity. I believe that natural immunity in most people resulted in immunity for about 2 to 3 years. So I'm not quite sure that if another infectious variant evolve, it can easily be spread in human population as others. For example, when Alpha was first recognized in Iran, in only 4 weeks, it completely displaced the classical variant. After that, Delta in only 3 weeks displaced the Alpha. But Omicron couldn't do this to Delta yet, because it is propagate with a lower speed than Delta. That's not because the Omicron is not more infectious. That's because the human bodies resist to get Omicron, and the virus can't easily propagate among population,
Yeah. So even though the virus is more intrinsically transmissible,
yes,
it's actually being impeded by the immunity that's enjoyed by the population, and that's slowing the spread down. So I'm thinking that if this pandemic will only become a problem again if there was a variant which outcompeted Omicron and was more pathogenic. And is it possible that could happen, that we could get a new variant which is very transmissible and yet still causes severe disease? Or does the fact that it's more transmissible mean the disease is likely to be less severe?
I can't completely say that it's not possible. It is possible, because a variant might evolve that escape immunity as well as more infectious. So the tragedy starts again. We had this problem with influenza. Influenza virus always change, and always new pandemic can exist. So it is completely possible. But the nature of coronaviruses is different from influenza viruses, because variation in influenza is much higher than coronaviruses. Coronavirus families are among the most conserved sequence families in RNA viruses, because the enzyme that is responsible for their replication has this proofreading activity that can edit mistakes, but influenza doesn't have. Another reason is that influenza genome is segmented, and when 2 influenza virus from 2 different species goes into a cell, this piece of genome can displace between them, and a new progeny can be produced that might be different from their from their ancestors, from their parents, and it can propagate to the population with new
It's almost like a sexual reproduction, isn't it? You get genetics from one virus mixes up with another.
Yeah, mixes up.
Does that happen though? I mean, does that happen with coronaviruses? Because this Omicron variant, does seem to be, it's not like the Delta, it's like something completely new, isn't it? Do you think there's been, can you get mixing of the RNA of coronaviruses if a host is infected with two different types of coronavirus at the same time?
Actually, yes, different theories exist about it. Some strange, some bizarre theories, and actually it might happen in nature. It might happen by a person, in a laboratory. I don't know actually what happened, but the number of mutation are very interesting. It has a lot of bad mutation inside, and it seems that they are sorted accordingly, to a logic together, So actually, I can't answer this question exactly, but it's very strange. We didn't expect that during this short of time, this much of variant produced during the pandemic, Personally, I didn't expect that.
Yes, I mean, in January 2020, what we were saying is, well, the influenza virus doesn't self-correct, therefore you get huge amounts of mutation and it changes overnight. Whereas coronaviruses, because they do have this self-correcting enzyme, they tend to be genetically much more stable. But here we are, we've got all of these variants. I mean, I guess one point is the virus has just been so prevalent. There's been so much reproduction. There's just been a greater number of opportunities for the virus to mutate. One question I get asked lots and lots, is it possible that a coronavirus can merge its RNA with a HIV, human immunodeficiency virus?
Yes, people talk about that. Actually, I don't actually know. It's I can't. In science, you can't easily deny something or accept something. It has to be proven.
Yeah.
But it's possible. It's possible because the RNA, if they co-infect someone, some genetic material change might happen. But as a scientist, I don't know about the rate, and I don't know about the likelihood of that happening.
So we can't say it's theoretically impossible, but we don't actually know of an instance where it happened so far.
Yeah. Because during the evolution of coronaviruses, some coronaviruses has this protein, hemagglutinin esterase fusion protein. Some believe that this protein similar to this protein exists in influenza C virus. Some scientists believe that the recombination between coronaviruses and type C influenza viruses has resulted to obtaining of this protein from one of these species. So during natural evolution, this event might happen, and I can easily deny the possibility.
Yeah. Is it possible that there could be another pandemic with a coronavirus or an influenza virus or a mixture of those two, or a virus we've never heard of next year that could have a really high mortality rate? Is that possible?
Yes, it's possible. Yeah, possible. It is possible. We have to prepare for the pandemics, because almost 5,000 species of coronaviruses are exist in bats, and we have recognized 500 of them. And always it's possible that they can change and they can obtain this possibility to transmit to human. So we have to be ready for other pandemics due to coronaviruses and other respiratory viruses. So it's not a good news for society, but we have to prepare to a better response, faster response, and prevent the disease to propagate. The experience of COVID-19 showed that we had a lot of problems in encountering a pandemic like that,
So it's possible that there could be a really severe pandemic anytime. Would it be, could we say that this is almost certainly going to be zoonotic if there is another pandemic?
I think because of the nature of the coronaviruses, because they exist in other species, it is highly possible that if it exists, it will be a zoonotic one, you know. Especially bats are a real bad host for coronaviruses. Bats, they have a lot of coronaviruses in them, and when people get close to bats and hunt them and bring them into the human population or society, it might be very dangerous.
Yeah. So basically what we're saying from virology is that there could be another pandemic that could be as potentially as infectious as Omicron.
Yep.
So it could go around the world, but potentially as deadly as Ebola. That's not a theoretical impossibility, is it? It is.
Yes, that's not true. But we were very fortunate that the pathogenicity of SARS-CoV-2 virus was not very high in comparison to many other viruses. And if it was, the number of mortality was much more higher, and the human nature was, threatened.
So have we been a bit lucky this time, really?
Yeah. Yes, we were. Have to be thankful. Yeah.
Yeah. So I mean, in virology, it's not impossible that you have a very pathogenic virus, very transmissible virus that, I mean, I don't want to be dramatic here, but it is scientifically possible that the majority of the world's population could be killed by a viral pandemic.
Sure. Yes, it is. Unfortunately, it's completely possible. And one of the threats to human being is the viral infection. So we have to be more prepared. We have to be more ready to reaction against them. And we need a very common sense for defending against viruses. In this pandemic, we showed that we are not ready to encounter pandemics. Some countries got their second, third, fourth shot. In some part of the world, vaccination rate is very low. And with this type of action, we can't assume that we are prepared and ready for another pandemic. We have to be more coordinate together. We have to work as orchestra when we are countering to
And I'm not too optimistic that that's going to happen anytime soon, unfortunately. So I mean, every year there's outbreaks of bubonic plague caused by Yersinia pestis, the bacteria. Yeah. But it's not a big problem because we treat it with antibiotics. Yeah. So is the future to stopping a pandemic having a very good quality antiviral drug where we could just clobber, just eradicate so just like we can eradicate. So in India, there might be half a dozen cases of bubonic or pneumonic plague. No problem. Treat it with antibiotics. That kills the bacteria, stops the person from being infectious. Is there any hope at all that we could have such a competent, comprehensive antiviral drug?
Yeah, we have to be hopeful, or we always have to be hopeful. We always have to work to make new antivirals and better antivirals. But there's a fact. Making antiviral is much more difficult than making antibiotics, because viruses are very simple. They replicate into our cells. So you have to find a compound that has this selective toxicity to inactivate the virus and doesn't harm the cell. So it's very difficult. Another problem with respiratory viruses is that in most of the case, the harm to the lung and to our body is not produced by the virus itself. In ARDS, or acute respiratory disease syndrome, most of the time it's the immune system, it's the immunopathology that results in ARDS. So viruses came to our body, it runs some processes, it hit some processes and then run. And after that, the processes that started act like a cascade and resulted in a bad situation. So when you eliminate the virus with antiviral, the outcome might not change very much after the cascade is run. So it's very difficult. in antiviral, I believe that if the antiviral to a respiratory virus starts in short time after the infection, it might be useful. But if it is late and the harm is produced and the people has this ARDS, it might not be very effective, you know.
I've got another.
Most of the
Sorry, yeah, I've got another question that's really quite unfair, but you're doing so well so far. So the immune system is this amazing structure. It can recognize 8 or 9 billion different foreign epitopes or antigens. It's just incredible. So why does it make the simple mistake of overreacting and killing the person who owns the immune system when it's exposed to a simple virus? Why is the immune system not better designed than this, that we don't get this overreaction?
Yeah, actually, the response to some pivotal questions might be very difficult. So I'm not an immunologist. I know immunology as immunovirology, we human don't know all aspects of the immunology. You know, we don't know a lot of things. But I believe that the responses sometimes they are out of control. In hepatitis B is like that. The hepatitis B virus doesn't harm to the liver, it's the immune system that attack and resulted in fulminant hepatitis. The same situation exists in ARDS with many viruses like influenza and SARS-CoV-2. I always example this like, imagine that the city of a country is occupied by some foreign forces. Immune system has 3 choices: come to a city and reasonably defend the city and make them go out. It can no reaction. It can do no reaction, so viruses can remain and propagate, and the city is occupied. Sometimes the reaction is so big, they attack the city with a lot of bombs, a lot of reactions, and even the enemy is killed completely, the city is destroyed completely. The same situation is occurred, yes, in fulminant hepatitis and in ARDS. Sometimes when a process starts and the cascade resulted in a disaster. That's why doctors give immunosuppressor drugs to the patient who experience ARDS at the early phase and try to oxygenate the liver and make the result better. You know, that's not all because of viruses, and that's why maybe because in children the response are more controlled because they don't have this actually complete immune system. They don't overreact, and the disease is milder in them. You know.
Right. So children are actually protected to some extent by their immuno-incompetence.
Kind of, or the doesn't have this rigid immunity that act, you know, in an uncontrolled way.
Yeah. And this is what we saw in the 1918-19 pandemic, where young adults were dying more so than people, you know, my kind of age, and then older people were dying as well.
Yeah.
So you know, we've seen this before.
Yeah. The situation in influenza might be different. You know, sometimes the virus can produce damage to the lung. Measles can produce damage to the lung. Influenza can produce damage to the lung. Some kind of hepatitis viruses can produce damage to the liver. But it completely depends to the virus and the type of immunity that activated against the virus and in different pathophysiological situations. You know?
Now, Omicron seems to be sweeping through Africa at the moment.
Yeah.
But the reports that I'm getting from colleagues in Africa is that there's not a lot of people getting sick. We hope it stays that way. But the vaccination rates in some areas are 2, 3, 4, 5%. You know, it's essentially nothing. So do you think that there could be cross immunity from other endemic viruses or other pathogens or even parasites in Africa that is protecting people against Omicron and indeed the other SARS coronaviruses as a form of cross immunity?
Yeah. It's one of the possibilities. Genetic markers are another possibilities. But personally, I believe that climate and weather should be more studied in different pathophysiological and situation. I believe that the climate has very huge effect on the severity of disease. Some disease are severe in hot climate. Some of them are severe in a colder climate. But I think we have to consider all these parameters to find out what's actually the main reason for that. Why the severity of disease in Africa, in Afghanistan, in Iraq is less than many other parts of the world with a lot of amenities in medicine. So the climate might be a very important contributing factor, and as you said, response to other pathogens might participate in these reactions. Good.
Fascinating. Now, I do have. Have you got time for a couple more silly questions?
Good. No.
How many viruses are there? How many types of viruses are there in the world?
There are a lot of viruses in the world. In human, about 300 different viruses are recognized. In other species, different viruses are recognized as well. In bacteria, in fungi, in algae, in insects, plants, every single organism that you study has a lot of viruses. So it seems that viruses are the most abundant creatures on Earth, but we know very few of them. About 4,000 viruses, 4,000 viruses are recognized by human, but we believe that they are very huge, both in terms of diversity and the amount, the number.
So we know about 4,000 types of virus. There could be millions of different types of virus. Yeah. So like humans have one set of viruses that can infect them. Dogs have another set of viruses that can affect them. Lizards have another set. Plants have another set. Bacteria have another set. Fungi have another set.
Yeah.
So there's like, and normally, unless we have spillover of zoonotic infection, the viruses generally stick to their own particular species. So every cell type has got its own subset of thousands of different viruses that can infect it. Yeah. This is just mind-blowing.
Yeah,
the complexity of this ecosystem.
Yeah, definitely. And some of them are useful. We use some of them for good reasons. Some of them are used as antibacterial. Phaage therapy is a science, when some resistance exists towards some antibiotics, microbiologists use bacterial phage to defend against some pathogenic bacteria. They might be useful even.
So we need to learn to use viruses as a species to our advantage.
Yes, sure.
Yeah. So if you go outside into a natural environment, you take a handful of soil, that's going to contain lots and lots of viral particles.
Yeah, more than 10 to the power 11 viruses might exist in 1 gram of soil.
10 to the 11. So that's a one with 11 noughts on the end.
Yeah.
And this is everywhere, all around the world.
Yeah, all around the world. Yeah, everywhere. Yeah.
And all these specific types for all the specific cell types. So mushrooms have their own type of viruses.
Yeah, definitely. We know very few viruses. The viruses that infect human and some animals and some insects, but we don't know absolutely most of them. And they're around us. They can do a lot of things that we are not aware of them.
So the assumption is that all of these millions of types of viruses and untold numbers of individual viral particles are necessary for the functioning of the ecosystem that we enjoy.
Yeah, sure. They can control the population of some insects, some plants, and they have some effects on the ecosystem like other organisms in the ecosystem. So they might be useful for us. And human pathogens are the viruses that we mostly focused on them, but there are a lot of them that might be useful, you know.
So the ecosystem on the Earth is a lot more complicated than we thought. Not only as the plants, animals, fungi, and bacteria, there's untold numbers of viruses that do things that some of the things we know about, and I guess many of the things we don't know about.
Yeah, sure.
Now, are the viruses in the sea as well? There's marine viruses.
Yes, the same amount of viruses, 10 to the power of 11, exist in 1 milliliter of in some oceans, you know. They're all around us, and fortunately only a few of them can infect human.
Yeah.
You know?
Yeah.
Almost 300 viruses can infect human, yeah.
So
that we recognize, you know.
So the assumption is that without all this massive diversity of viruses, the ecosystem would not be a functional ecosystem to support human life.
Yeah, sure, definitely.
So we complain about pandemics, but we actually need viruses.
Sure. Yeah, to control the population of other organisms to make the ecosystem act normally, you know.
Yeah. So again, I know there's different theories on this, but where did all these billions of types of viruses come from?
Actually, we don't know the origin of viruses. Some believe that they are the first form of living on Earth, the first form of living produced from an RNA, and this RNA is a virus. Some believe that they are the deteriorate form of intracellular parasite like Rickettsia, Mycoplasma, and other bacteria. And some believe that they are autonomous nucleic acids that come from our cells, living cells, and can propagate autonomously. And there are some bizarre theories that says viruses came from other planets with some kind of
the panspermia idea.
Sure.
I don't buy the one about coming from bacteria, because there'd have to be billions of different types of bacteria. Simplifying into viruses, and so
some
we basically don't know where they came from, but we do know that we do need them.
Yeah, but some viruses might evolve differently, you know. There is a difference between viruses with DNA as genome and viruses with RNA as genome, and the viruses that use both of them. We actually don't know the real origin, but the origin might be different. We call them virus, they might be different organisms with different histories, you know.
Could be completely different, But the genetic coding is the same between the RNA and the DNA viruses. The codons, the genetic sequences code for pretty similar, generally amino acids, so that they are related in some way, aren't they?
All forms of living have these genetic codons. You know, it is the meaning of codons is almost similar from viruses to worms to insects to human. You know, so
all life on Earth, whether it's a simple RNA virus or us, is working on the same chemical genetic code principles.
Sure. But they have some specialties for their own, you know. Some viruses can use RNA as genome. It's quite different from other form of living creatures on Earth, you know, like bacteria and eukaryotic cells use DNA, but viruses can use RNA, and DNA. Some of the replication strategy for all of them might be very different from each other. So we have to study every single viruses in a separate way to understand how they replicate. They normally sometimes do their special things for themselves, you know, in terms of replication, propagation, and acting inside the cell.
But in every case, all the viruses that we know about can only evolve and reproduce inside other living cells.
Yes, sure. All viruses.
Yeah, yeah.
They are obligative genetic parasites.
Yes, yes. It's the only way they can reproduce. Yeah, so that would kind of make it difficult to have a virus before we had the first cell, wouldn't it? Yeah, we've got a real problem there. Let's hope the evolutionary biologists are working on that. Now, the last question I've got is it true that there is viral genetic material inside my cells and your cells?
Yeah, it's completely true. Inside our genetic material, there are some kind of retroviruses that we call them human endogenous retroviruses. And the endogenous retroviruses exist in all eukaryotes. And if you consider them, sometimes we assume that about 10% of our genome is formed from that kind of materials, human endogenous retroviruses. And there are some genetic materials into our genome that are the deteriorate form of that viruses. We call them retrotransposons. And if you consider all them together, about 30% of our genome is produced from that kind of viruses. So they are, yeah, endogenous genetic material in us. We don't know actually, are they inserted into the genome of our progenitors, or they are the genes that the viruses evolved from them. It's
So
very difficult.
I inherited a lot of viral DNA from my parents.
Yeah.
Now,
And
this 30% of the genome, does this 30% of the genome code for structural proteins? And does it code for functional proteins like enzymes? Do we know that?
Yeah, sure. They do produce some enzymes, reverse transcriptase enzyme that exists into our cells, came from this genetic material, these viruses, and they act in our body. Many different biological roles for them has proposed. We don't know actually. They are really mysterious. Some biological behavior, some acts are related to the endogenous retroviruses. And I think when they exist in our genome, they have to do something for us. If they were junk during the evolution, they removed from our genome. So they are there to do something, and maybe if they didn't exist, we couldn't exist as well.
Oh, weird. So I mean, do we know if any of them are structural genes for structural proteins?
Yes. Some of them, yeah. My knowledge about them is not very much, but sometimes they produce even viruses. In some species of animals, it is shown that they can come out from from the genome of the organism and produce viral particles. Some disease is assigned to them. Some biological activities is assigned to the, and retrovirology and endogenous retrovirology is a discipline in virology that many scientists in the world are involved in the study with them.
So vertebrate life is only here and only works because of the incorporation of viral infections, essentially, in the past. So we are part virus then.
Yeah, sure. Yeah, it's strange, but it's true.
Life is just so complicated and amazing, isn't it? It's just, and yet here we are, alive and conscious and talking about it. It's absolutely crazy. Behzad, thank you very much for that. Fascinating talk. Yeah, my mind's still spinning with this idea that I'm 30% virus, but and the complexity of the viral ecosystem is just amazing. But I think if science doesn't produce a sense of awe, a sense of amazement, then we're not communicating properly.
That's true.
Because the nature of reality is just beyond the comprehension of any individual. That's for sure. And yeah. Great, Behzad. Thank you very much for that. I'd be interested to see what feedback we get on these talks. So thank you for taking so much time out of your busy schedule.
Thank you so much, John.
Thanks.
Thank you for your time. I enjoyed it.
Yeah, thank you.